All right, this is part two of our cellular respiration lecture. We're going to be talking about the actual process of cellular respiration, which is here in front of you. We are going to be doing the transfer of glucose, which is C6H12O6, to carbon dioxide and water in the presence of oxygen. This is kind of like burning the molecule of glucose. Remember in class, I actually burned a package of sugar, and you saw that fireball that comes when you burn it? The cells do that, but in a micro scale, a control scale, in a series of biochemical pathway steps, facilitated by enzymes that are lowering the activation energy and speeding out the process. And throughout the process, this energy is released in a series of oxidation reduction reactions in a controlled manner, in which the cell can actually trap that energy into ATP in a very efficient manner. So that's cellular respiration. Now, this is going to be happening inside of mitochondria. Mitochondria is basically a living organism living inside all eukaryotic cells. It qualifies for all the categories of life. It self-replicates. It has DNA. It has its own internal stable environment, which it tries to maintain. It can make proteins on itself. It can replicate its own DNA. And basically, this is the structure of it. It's got an outer membrane and then an inner membrane. So even a double membrane endosymbiont. And in between, it has an inner membrane space, which is going to be important because it's a very acidic environment full of hydrogen ions. We're going to talk about why that's important when we do the ETC. And then it's got this swimming pool of cytoplasm-like material that's called the matrix, where you would find ATP synthesis particles, enzymes, ribosomes to make their own proteins, granules full of proteins that it makes, and its own DNA as well. So mitochondria is technically alive. Now the theory is, of course, that at some point early in the evolutionary process, mitochondria as well as chloroplasts were imported into the cell by phagocytosis, but that instead of destroying these mitochondria, the cell established a symbiotic relationship where the mitochondria actually helped the burning of glucose molecules and facilitated the energy process of the cell, which was why the cell maintained the mitochondria. And so now all eukaryotic life forms have mitochondria, and you can't really find mitochondria anywhere else. So, but it's definitely kind of like its own endosymbiont. Now, another thing we're going to talk about before we move out of the mitochondria slide is the idea that the matrix provides the chemical reaction environment for the citric acid cycle, the Krebs cycle. So it's got the chemicals necessary to happen for the citric acid cycle to happen. And the cristae are the membranes in charge where the ETC is going to happen, which is actually what produces the most energy. Which is why these members are all folded. Because if you had a mitochondria that had a shape like this, where the inside membrane was just one circular membrane, and you compare that with another mitochondria that actually has ups and downs on that membrane, and you have all of these foldings, clearly the mitochondria on the left side will have a greater area. If you were to run around this track here, instead of running around this track, definitely you will run a bigger distance on the left side because of the folds. So what the folds are doing is maximizing the surface area for the chemical reactions to take place inside the mitochondria, allowing for more ETC proteins to be available and a greater efficiency at producing the proteins and the energy from the cellular respiration, which is why this evolved. Basically, a mitochondria that had folds was better than a mitochondria that didn't, and it survived better. So that is why it evolved that way. So now let's talk about the actual process of cellular respiration. It happens in 4 steps. The glycolysis, which is the first step, followed by the grooming step. The grooming step will be the step of transfers between the cytoplasm into the mitochondrial matrix. Then at the matrix, you will have the Krebs cycle. And then at the membranes, between the inner and outer membrane, you will have the ETC. Notice that during the glycolysis and Krebs cycle, ATP is produced through substrate level phosphorylation. Now what that means is that ATP is made through enzymes and chemical reactions in liquid environments through a series of biochemical pathways. But during the ETC, it is done through oxidative phosphorylation or chemiosmosis, in which the enzymes are involved are actually a little turbine that spins to create ATP. It's quite fascinating. And we're going to be talking about that. But all of that energy is generated based on the electron carriers produced during the glycolysis and the Krebs cycle. So you see that these electron carriers are going to be going into the ETC to create the energy for the ETC to take place. So let's talk about this, how this actually happens. So it all starts with glycolysis. Now, glycolysis is what all glucose molecules undergo this process. It's anaerobic, it doesn't require oxygen, and it happens in two major steps. First is the investment phase, where a glucose molecule is charged with phosphate groups donated by 2 ATPs, so this actually consumes 2 ATPs, all right? And is going to go ahead and create a charged molecule called pGAL, or glucose, a charged glucose. Now, this molecule will then be split using the energy that was charged. It will be split to create half of that, which are called pyruvates. Now, notice the pyruvates are 3 carbon molecules, and glucose is 6 carbon molecules. So you did cut it in half. So you're going to make 2 pyruvates out of each glucose. So whatever happens from now on is going to be doubled because of this, because you made 2 pyruvates from each glucose. Now notice that doing this split process here, you actually create 2 NADHs and 4 ATPs. So even though you had to spend 2 ATPs, you end up making 4, which gives you an overall net ATP of 2 ATP after glycolysis is over, in addition to the 2 nitrox up here. So you have these things. Now, before we continue, let's do a little talk about these electron carriers that are going to be doing the job of carrying the energy during this process. So you have ADP, and you have NADH, right? And you also have FAD. And all of these things are uncharged versions of the carriers. Now, when these things get reduced, so when these things get reduced, they receive electrons, they become charged, like ATP, right? NADH, which is the charged version of NAD, and then FADH2, which is the charged version of FAD. So these things will create the charged version. Now, when you uncharge them, you oxidize them, and the electrons are lost, and they become uncharged again. So remember that every time you see ATP being formed, that means ADP was consumed. Every time you see ADP being formed, it means ATP was consumed, and vice versa. So this is an oxidation reduction that keeps happening throughout the cycle. Now, if you actually look at the bottom here, you will see that this is the actual breakdown of the process of glycolysis. And as you can see, it's much more complex than the simplified investment versus payoff phase thing. And clearly, it takes a lot of enzymes and a sequence of biochemical pathways to finally charge the glucose molecule. So as you can see, you go through several steps until you finally end the investment phase here. And look at the ATP being consumed here and here, right, to get to this charged glucose. And then this glucose will split into 2 molecules, which are the pGALs, which will then undergo a process that's going to produce 1 truck here and 1 ATP here per molecule, and there has you got the other ATP. But since this happens twice, you're going to get 4 ATP and 2 NADHs out of this process, giving you an overall of 2 ATP and 2 NADHs. So that is the glycolysis. Now, once glycolysis is over, it all depends on whether or not the cell has oxygen. If the cell has oxygen available, and this is regulated through enzymes, the presence of oxygen activates the pathway that sends the cell into what's called the grooming phase. Now, in the grooming phase, basically what's going to happen is the pyruvate produced during glycolysis is going to enter the mitochondrial matrix. And in the process, it's going to be converted into acetyl-CoA. Now, CoA is basically an enzyme that's going to facilitate the initiation of the Krebs cycle. And acetyl is a 2-carbon molecule. So you're going to go from a 3-carbon molecule here to a 2-carbon molecule there, which means carbon dioxide was produced. And since you have 2 pyruvates, you're going to make 2 carbon dioxides. All right? During this process, you also generate another 2 NADHs. So by the end of this, you have 4 total NADHs. Unfortunately, this process actually costs 2 ATP. And that's pretty much what the mitochondria takes out of it, is the mitochondrial tax. The grooming phase requires 2 ATP. It's basically when the actual insertion of the coenzyme into the acetyl molecule requires ATP. So let's see how that actually works when we do the Krebs cycle. I'll mention that again. But the grooming phase is just the initiation of the Krebs cycle, all right? Now, in the Krebs cycle, basically, the acetyl-CoA that was created is going to undergo a series of metabolic processes where the 2-carbon acetyl-CoA molecule combines with a 4-carbon oxaloacetate molecule to go ahead and become a citrate, or citric acid, which is why it's called either Krebs cycle or citric acid cycle. Krebs cycle after the scientists that discovered it. Now, the citric acid cycle will then continue, and through a series of chemical reactions, the citrate is going to go back to oxaloacetate. So you're going to go from a 6-carbon back to a 4-carbon molecule, which means you're catabolizing it, and in the process you're producing 2 carbon dioxides, another 3 trucks, 1 ATP, and 1 FADH electron carrier. Now, since you start with the 2 pyruvates, you're going to make 2 acetylcholates, and you're going to do all of this times 2, which means you're going to make a total of 4 carbon dioxide here, 6 of the natrucks, 2 ATPs, and 2 FDHs. So, where are we at after this? So after glycolysis, you had a total of 2 ATP and 2 NADHs. After grooming, you lost the ATP, so you're down to 0, but you gained 2 more NADHs, so you got up to 4 NADHs. After the Krebs cycle, now you got 2 ATP back from this, and now you have 6 more NADHs, so you're up to 10, and now you have 2 more trucks, so you have 2 FADHs. Now, those 2 ATP, the cell's going to go ahead and use it, but it's those trucks, those 10 NADHs, and those FADH electron carriers, I call them the electron carrier trucks, which have to be converted to the battery, which is ATP, which is what the cell actually needs to use, which takes us into the ETC. But before we do that, let's look at the complicated version of the Krebs cycle. So as you can see here, again, a series of biochemical pathways, and it's actually including the grooming here, because you got the pyruvate that is going to go ahead and combine with the acetyl-CoA, and that is going to require that ATP we talked about to make the acetyl-CoA down here. Now remember, this coenzyme, it's all it's doing is facilitating the process of combining the oxaloacetate, which is that 4-carbon molecule, with the acetyl-CoA, which is the 2-carbon molecule. Also notice the pyruvate is 3-carbon, and then a carbon dioxide is released. That's how you get to 2 carbons. And then that's going to combine with the citrate, the oxaloacetate, to make it the 6-carbon citrate. And then through a series of chemical reactions and enzymes, and all of this is catabolized, you're going to be changing these molecules. And throughout the process, you're going to make electron carriers here, 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 there, and you also make carbon dioxide here and here. So when all is said and done, you make what I described about in the beginning of things. Also notice here that you actually are not making ATP, you're making GTP, which is another kind of electron carrier. But this GTP is going to immediately be converted into ATP. It's going to later on. So you're still going to make ATP out of the citric acid cycle. And this is right here is what's going to go ahead and become the FADH truck. All right. So very complex, but you don't need to know that unless you want to go crazy and memorize it. But it's definitely not necessary. So from here, the cell is going to go on to the ETC. Now, during the ETC, basically what happens is that the trucks produced during the Krebs cycle are going to be powering up the reactions of 3 proton pumps, which are going to be putting hydrogen ions from the matrix into the intermembrane space, creating a low pH of 4 outside the matrix and a pH of 8 inside the matrix. So what's happening here, the electrons that are stored inside NADH and stored inside of FADH are going to be powering up those pumps. Now notice that NADH actually power all 3 pumps, while FADH is only going to go ahead and power 2 of the pumps, so that the NADH is going to end up making more power than FADH does. I call it the fat truck, doesn't work out as much. Also notice that at the end of the cycle, an oxygen atom right here is going to be accepting these electrons, and then going to be reduced to form water, and that's where the water is produced during the cellular respiration process. Now, what does the cell get out of this? What you're creating here is an electron gradient, proton gradient, or electrochemical gradient, where you have this proton motive force of a lot of protons wanting to get back by diffusion inside. But the membrane is selective, it doesn't let that happen. So only in the presence of a channel, such as the ATP synthetase here, will these protons be allowed to go back inside. But as they go back inside, they have to spin this turbine-looking thing. And every time they spin this turbine, you're going to go ahead and produce ATP. So that is how the ETC works. And you can see this actually a very complex process that takes a lot of chemical steps. But when everything is said and done, these things will produce ATP. And we're going to continue from here in the next lecture, where we're going to do an overview of a summary of the cellular respiration process.